This week we're going on to Chapter 17. And Chapter 17 talks all about how we identify and diagnose specific pathogens, especially when someone presents with a disease and we need to identify exactly what the disease is. There are three main methods we use to diagnose infections and identify pathogens. The first method is the phenotypic method. So phenotype is actually visualizing characteristics of a pathogen or of an organism. So, for example, we might look at it under the microscope. We're actually looking at the shape. We're looking at it if it's gram-negative or gram-positive. We're looking at the size. Or we might do a macroscopic examination. This means we're looking at it on like an agar petri dish. We want to see what the colonies look like, the color, the shape. Are they growing on certain selective or differential media? Lastly, we might look at some physiological or biochemical characteristics. So we're really looking at, does this bacteria have certain enzymes? Is it sensitive to certain antibiotics or resistant to antibiotics? So this can tell us very specifically maybe the strain of the pathogen that's causing the disease. Phenotypic methods are most commonly used for identifying bacteria and some eukaryotic infections, like maybe some worms or fungi or protozoa. Things that are big that we can actually see under the microscope. We would not really use phenotypic methods for identifying viruses because we can't cultivate viruses in the lab and we can't see them under traditional microscopes. You need an electron microscope for viruses. So phenotypic methods, these are typically used for the bigger guys, bacteria and eukaryotic microbes. We might use them if we need really quick results because we can do a microscopic slide in like 2 seconds. So if you need like a quick gram stain, you could do it then. Or if we actually need to grow the bacteria because there's not a lot in the sample, we might have to cultivate them on media using macroscopic examination. But otherwise, phenotypic methods are not the most best or not the most commonly used techniques because it is kind of slow to have to grow the bacteria out on a plate. As you guys know, it takes several days. Biochemical methods are also slow. They can be expensive. The next main group is genotypic methods. So in this case, we're looking at specific DNA or RNA of the pathogen or its genetic makeup. Genotypic tests are much more commonly used, especially in this day and age, because they can be pretty cheap, they can be pretty fast, and they're extraordinarily accurate. We can pinpoint exactly what the pathogen is by looking at its genetic makeup. And of course, it's not always necessary to culture the microbe first. So again, this can provide us really fast results. The third group is immunological testing. This testing relies on antigens and antibodies. Antigens are specific surface structures found on the pathogen. Often they are a protein associated with the pathogen on its surface, like its pili, its flagella. Maybe it could be its capsule, which is a polysaccharide. Antibodies are proteins made by our human immune systems that will specifically attach to antigens. And when an antibody binds to the antigen, this targets the antigen for destruction by the human immune system. So both genotypic and immunologic testing are very frequently used. They're very fast, rapid, and they're much more specific in identifying the pathogen than phenotypic methods. Also, these two groups of methods can be used for identifying viruses, as well as bacteria, eukaryotic microbes, pretty much everything. One of the most important things to consider is when you collect the actual specimen for examination. And this is something you guys will probably work with directly. So depending on where the infection is, different types of samples will need to be collected. So for example, if someone has a lower respiratory tract infection of some sort, you will need to collect a sputum sample. So for this, the main consideration you have to think about is that the normal flora in the mouth, especially in saliva, contains a lot of bacteria already, a lot of normal flora bacteria. So how do you differentiate normal flora bacteria from the pathogen? So for example, you might have to have the patient brush their teeth or swish with some type of mouthwash prior to sample collection to try to eliminate as much contamination as possible. You'll never eliminate all of it. And then for a sputum sample, you would have to have the patient cough up a sample. From there, the sample needs to be stored properly, either refrigerated or in some cases it can't be cold, it cannot be refrigerated. It always depends on what the sample is. So for example, a cerebral spinal fluid sample from a spinal tap should not be refrigerated, and it should be processed immediately. While some samples it may be okay to store overnight, others it's definitely not okay. Another example is collecting a urine sample, like you're testing for a UTI. Usually when collecting a urine sample, you want to do what's called a clean catch, which means the patient cleans the external genitalia and then they urinate and they collect the sample midstream so that any normal flora bacteria that could potentially contaminate the sample is eliminated. So it always just depends, and you always want to, no matter what, remember to maintain aseptic technique to help eliminate as much contamination as possible. Sometimes it's really hard to differentiate between the normal flora and pathogens. It always depends on the sample. So for example, in urine, there's always going to be some E. coli that may get in there just from the normal flora. So if you see a few E. coli cells in the urine, okay, that's normal. But if you see hundreds or thousands of E. coli cells, then that's not normal, and that could probably indicate a UTI. This chart, which is also in the textbook, is really great. It just kind of tells you how to collect the sample, any additional comments on storage, things like that. Another thing to really keep in mind is if you suspect a certain pathogen, it might need to be stored a certain way. So for example, if you suspect someone is infected with Neisseria gonorrhea and you do a genital tract sample, you need to store the potential Neisseria in a microaerophilic environment. So maybe a high CO2 atmosphere, since it is a microaerophile and it doesn't tolerate very high oxygen. Other samples, maybe deep tissue samples, need to be kept totally anaerobic. And they actually make anaerobic serum vials and bottles to store anaerobic samples in. As I mentioned, once the sample's taken, there's tons of different tests that could be done, either phenotypic, genotypic, or immunology tests. If a test is going to provide you pretty immediate results, like you've taken the sample and you can immediately get results from it, this is called a direct test. So for example, looking at it under the microscope, it takes like 30 seconds, it's quick, it's a direct test. If you're looking for certain antibodies or antigens in the sample, it's very quick, rapid test. Or there are some genetic tests that are also direct, very rapid, you don't need to cultivate the organism. If you do need to cultivate the organism or isolate it in order to identify what it is. So for example, if there's a cerebrospinal fluid sample that has very small quantities of bacteria and you need to grow more of them in order to identify what it is, you have to put it on a petri dish, then these are not direct tests. We usually categorize them as culture or isolation tests. So for example, if you were doing a antimicrobial sensitivity test, a Kirby-Bauer test, you would have to grow the microbes and then put the discs with the antibiotics on there, and that takes several days. If you had to grow the bacteria in an animal, because a lot of bacteria don't grow in the lab on agar, you have to actually put them in a living organism. So if there needed to be an animal inoculation, that would take several weeks in order to identify the pathogen. So it always just depends on what we're working with. If we have a general idea of what the organism is, we say it's presumptive. So for example, if we knew the potential genus, if we had a good idea that our pathogen was Staphylococcus of some sort, but we didn't know what, this would be presumptive. If we knew exactly what strain and species that we were dealing with, we would say it's confirmatory. So for example, if we knew it was Staph aureus or methicillin-resistant Staph aureus, we knew exactly what it was, it's confirmatory. It's not always necessary to exactly identify or exactly confirm what the pathogen is. It always depends on a case-by-case scenario. So for example, if you had a young and very healthy patient, they were feeling maybe like a stomach bug and it kind of went away, but they still went to check up anyway, the doctor probably would say, hey, it's probably just, norovirus. We don't really need to find out exactly what it is because you're feeling better. The doctor can kind of just look at the signs and symptoms of the patient and let them go without needing to test. Other times there are things called point of care tests. These are kind of like rapid tests. They're almost similar to pregnancy tests. They're usually looking for a specific antibody against a disease. So for example, they make these rapid point of care tests for HIV now. They can actually use saliva to look for anti-HIV antibodies. If someone has antibodies against HIV, that means that they have been exposed to the virus. They also make these rapid tests for strep throat and other common diseases. The problem with these rapid point of care tests is they're not always very accurate. There's a lot of false negatives and false positives, which can actually be really dangerous. There are also some what we call in vivo tests. So in vivo means the test is actually done in the patient. So for example, when someone gets the tuberculin skin test, it actually is done in the patient, right? Or if someone gets an allergy test, they're inoculating a small amount of the allergen and looking for a reaction, that's in vivo. The majority of tests are in vitro. In vitro technically means kind of like in a test tube, but it doesn't have to really be a test tube. It just means not in the patient. Requires sampling and requires testing outside of the human body.